All right. Well, good morning, afternoon, or evening to anyone who's joined thus far. Thanks for coming to this meeting. For anyone who's attending this call for the first time, we basically have been holding this industry-wide call every month with companies that want to contribute to the cultivated meat value chain. And so the purpose of these calls is to really offer new or existing cultivated meat stakeholders an opportunity to learn from each other, especially with respect to certain obstacles and potential solutions that might exist along the value chain, ultimately, hoping to accelerate R&D and commercialization in the field. So if your group is interested in presenting on a future call, or maybe you're working with a company that could benefit from being on these calls, please do feel free to put them in touch with me directly, and we'd be happy to get them added or scheduled as a presenter. Pleased to welcome Guy Bushkin from MNDL Bio today, who's going to be sharing more about strategies to optimize recombinant protein production. So as always, feel free to drop questions in the chat throughout, and we'll get to those at the end of the presentation. So Guy, I will turn it over to you.
Thank you, Elliot. My name is Guy. My background is a PhD and a postdoc in microbiology and molecular biology. In the past 4 years, I've led research in 2 biotech startups, one in the protein engineering space and another in the strain engineering space. Very recently, I joined MNDL Bio as the Director of Product, Synthetic Biology, and I'm here today to tell you about our approach to protein expression optimization. So here's an overview of today's talk. I'll start by giving some brief background on challenges in recombinant protein expression. I'll then move on to describe MNDL Bio's approach to gene optimization. This will be followed by a couple of case studies to showcase our capabilities. And finally, I will say a few words about how MNDL Bio can help with research in the food tech space. Recombinant proteins are a cornerstone of biotechnology with applications in therapeutics, transgenic animals and plants, precision fermentation for synthesis of chemicals, including food ingredients, and protein production, such as growth factors for cultivated meat. So we can have delicious hamburgers like the one here on the right. Now, protein expression is a multi-step process, and I cannot list all the steps here, but I would like to highlight some of them to give you an idea of the complexity of it. We have to start by considering the conditions that the cells are facing, the nutrients availability, the temperature, the pH, the position in the cell cycle, and if we're expressing from a vector, the copy number. Then we have to think about everything that goes into transcription, promoters, transcription factors, RNA polymerases, and even DNA and histone modifications. Then there is everything that happens post-transcriptionally to the mRNA, splicing, localization, and RNA modifications, to name a few things. Then we have to start thinking about translation, UTRs, poly-A tail lengths, initiation, elongation, the availability of tRNAs, and mRNA stability. And finally, when we're interested in an active protein, we also have to think about folding, post-translational modifications, localization, including secretion, and protein stability or degradation rates. Now, when we are trying to model all of these processes, there are several issues that make this very difficult. First, factors controlling protein expression are often non-additive. Those of you with a background in genetics might know the term epistasis. This is when you have, let's say, 2 mutations or 2 alleles, each one, let's say, with a negative effect on fitness individually, but together they have a positive effect. So 1 + 1 does not always equal 2. It can be either more or less than 2. This is the meaning of non-additive. each of these steps can be bottlenecked by a downstream step. So it's not enough to optimize only one of them or to model one of them. You have to consider all of them. And all of these steps are organism and cell type specific. The second issue we face when trying to model protein expression is lack of direct measurements. So that makes it we have not enough data to model gene expression. Now, there are several challenges in biotech R&D, and protein expression is one of the main ones. This can affect biotech R&D in several ways. During screens, low expression result in reduced data collection. For example, suppose you're looking for a certain enzymatic activity and you are expressing variants of an enzyme in a 96 well plate. If you only end up with 40% of them expressed, you only collect the enzymatic activity for 40% of them. And maybe the highest activity was in the rest of the enzymes that just did not express, so you could never measure them. During scale-up and production, lower yields lead to increased costs because you get less product from each batch. Let's say you're doing fermentation, you're making some protein, you're getting less yield from each batch, that increases your costs. And as many of you probably have witnessed or even experienced yourselves, low protein expression can lead to prolonged timelines, increased costs, and that often results in project cancellation. If that happens in a startup, it can be fatal to the startup. So here in MNDL Bio, we say that synthetic biology was imprecise, costly, and limited. We are MNDL Bio. We provide protein expression optimization solutions. We are a spin-off from Professor Tamir Tuller's lab at Tel Aviv University. We have 15 years of experience developing models and algorithms for gene expression. We have developed hundreds of algorithms that were featured in over 160 peer-reviewed publications. We have multiple patents and dozens of successful collaborations, both within industry and academia. When people talk about gene optimization, they mean it in several different senses. One sense that people mean when they say gene optimization is codon optimization. When you do codon optimization, you take your recombinant protein sequence and you choose codons for the amino acids in that protein based on their frequency in the host genome, using a codon usage table such as the one here on the right. The issues with that are as follows. It is often inconsistent, meaning it works for some proteins, but it doesn't work for others. And it's not always clear why. It reduces a gene sequence to a series of single codons, ignoring higher order information. Maybe it's important to have a codon at a certain frequency at some position and a codon at another frequency in a different position to regulate the overall rate of translation, for example. And finally, it might eliminate rare codons in positions where they are preferred. For example, where ribosome slowdown is needed. Maybe you need to first allow for folding of one domain of the protein before proceeding to translating the rest of the protein. Otherwise, it ends up being misfolded. Another sense in which people use the term gene optimization is when it comes to DNA synthesis and gene synthesis. You go to the website of the gene synthesis company, you paste your sequence, and they do some kind of optimization. They look for GC content, and they usually remove homopolymers and low complexity sequences and hairpins. However, they are primarily concerned with the DNA synthesis itself, meaning the oligonucleotide sequence synthesis and its assembly into genes. And While some of these optimizations might result in increased protein expression, that is not the main concern, the main thing that they are trying to optimize. So here are a few ways in which MNDL Bio's approach to gene optimization is unique. We optimize not only the genes themselves, but also regulatory sequences, including promoters, terminators, and UTRs, and even the vectors themselves. We use a multidisciplinary approach based on biology, physics, and engineering, leveraging AI, biophysics, and more to develop advanced computational models spanning every step of the gene expression process, which can be adapted to any host. Now, you might still wonder, why do you need models to begin with? And the answer is the enormous search space. Let's consider a conservative case where you have a really short protein of only 100 amino acids long, meaning 100 codons. And suppose you had only 2 options in each of these positions, only 2 codons. Many amino acids have 3, 4, and even 6 options. But let's be conservative and suppose you had 2. That leaves you with 2 to the power of 100, which is about 1 and 30 zeros afterwards. Okay, it's an insanely large number, and you will never test all of the combinations yourself. You'll never be able to test it. So you must use models to reduce the search space. Our products are optimized DNA sequences for improved protein expression. And we have a track record of dozens of recombinant proteins that were successfully expressed in multiple hosts. So to dive a little bit more into MNDL Bio's approach to gene optimization by giving you a few examples, I would like to first highlight that we have a comprehensive set of models for translation. These models are host-tailored. They take into account the pool of available ribosomes in the cell. They look at rates of translation or stalling along mRNA. They look at mRNA folding, and they distinct between the initiation and elongation steps of translation. Some of these models are published and some are non-published. This is one example from one of these publications that I cite below, where the team was able to identify features in the 5' UTR of genes that are in correlation with high rates of translation initiation. This was done by looking at RNA-seq data from pull-down experiments of the small ribosomal subunit. Another way in which MNDL Bio's approach to gene optimization is unique is by looking at codon series. A gene is more than a series of individual codons. We use a set of algorithms for matching nucleotide strings in the target gene to those of the host genome to capture information not present in individual codons. And lastly, we also look at vector stability for long-term expression. So this is especially important for those of you who are in the precision fermentation space. As you may know, sometimes the culture is being overtaken by mutants who no longer express your protein of interest because of the associated fitness cost. We have models to balance the trade-off between high expression and fitness, and we can also couple the expression of a recombinant gene to an essential gene to reduce the likelihood of loss of expression. Let's go into a couple of case studies to showcase what we have done. In this example, this was a project with a company who expressed a protein in a tobacco plant. The baseline situation was that the product was already in the market. So the company already settled on an amino acid sequence. But most of the protein was inactive, despite high mRNA levels. We worked with them to optimize the gene, only the nucleotide sequence, without changing the amino acid sequence, to improve co-translational protein folding. In this example, the original sequence, set here to 100% activity, was compared to 12 variants that we provided them, and one of the variants had a 200% increase of inactivity over the unmodified sequence, or a threefold improvement. In another example, we looked at we helped to develop a biosensor by increasing expression of a reporter gene in E. coli. This was done in collaboration with an academic lab. They were interested in developing this biosensor for environmental monitoring. The idea is that when in the presence of some small molecule in the environment, the bacteria emit light, thus function as a biosensor. The baseline situation was that the previous biosensor version was active in the lab, but it was not viable for field application due to low luminescence signal. We designed 370 promoter variants and tested them. So you can see these variants here. Many of them had higher luminescence relative to the luminescence levels of the control, the unmodified promoter. We highlight here one of these variants. It's not even the most highly luminescent one, and it had a 10-fold increase of the in the modified promoter sequence over the unmodified promoter. So where does MNDL Bio fit in with alternative protein and cultivated meat? Well, there are several ways in which we can support research effort and production effort. We can use MNDL Bio's platform to optimize protein expression and support discovery efforts to increase yields and reduce screening times and collect more data from each iteration. We can help with your scale-up efforts. Let's say you had already settled on your amino acid sequence. We can help increase the yields by changing the nucleotide sequence and thus increase the profitability from each batch, or reduce the costs. Some areas where we can partner with you are anything that has to do with precision fermentation, let's say production of pigments, sweeteners, or emulsifiers, and anything that has to do with protein expression, such as growth factors for cultivated meat and alternative proteins. We have experience supporting protein expression in several types of yeasts, fungi, plants, microalgae, and bacteria. So the take-home message here is that at MNDL Bio, we have a comprehensive, multidisciplinary, host-tailored approach to gene optimization that is different than what currently is thought of when the term gene optimization is used. We use machine learning and AI to reduce the search space and provide solutions that are orthogonal to traditional methods, for example, mutagenesis. We optimize recombinant protein expression with applications in food tech, and we have a success record of dozens of recombinant proteins in multiple hosts. If you would like to partner with us and boost your protein production, contact us at guy@mndl.bio. Thank you, and with that, I will take any questions you may have.
All right. Well, thanks so much for that presentation. I think it's a good sign of a maturing ecosystem when we have new companies entering the field with such specific expertise that can sort of support really more sort of niche applications in the field. So thank you so much for coming and taking the time to present to us. Hopefully some of these folks working on some of these problems might reach out to you and help you troubleshoot some of these challenges. So appreciate the time, and thanks everyone for attending.
My pleasure, and thanks for the great questions, everyone.
All right, we'll see you next month. Bye.
Thank you.